My name is Matthew Jose. So today I'm bringing you something a bit different. Still part of pharmacology, but we're going to be looking at the different types of penicillins, different types of antifungal drugs, antiviral drugs, and so on. So here you can see the first. Before I start off with the video, I would just like to say this lecture slides is taken from my university, so it's not by any means created by me. It's created by Dr. Anna from the Department of Pharmacology within the University of Liverpool. Right. So now, before we begin, what we want to do is go back and think about who created Fleming. So you know Alexander Fleming. He was the first one to invent or find out about penicillin. So he was doing this when he was researching about streptococcus bacteria one day, and so he had and it was in 1928 when he came up with this invention. So what happened was he was researching on streptococcus bacteria, and he had bacterial colonies. So one strange day he found out that there was a mold. So here you can see that, and he investigated what was the speciality of that mold, and he found out that there was no bacteria presence there, which means all the bacteria around there had been killed. And then he subsequently went on to do his research and found out the molecule called penicillin. So penicillin is the family name. Antibiotics come under penicillin, right? So the main structure that you want to be aware of it as penicillins as a ß-lactam ring right here. So there's a square thing with the ketone sticking out. So it is aromatic in a sense if you look at it. And they have an R group. The R group is very important in the sense that it affects the stability of the enzymatic or acidic hydrolysis and any antibacterial spectrum. So what this means is that the R group defines what class of penicillin it will be. It also explains how acidic hydrolysis can take place as well. So in terms of some bacteria which are resistant to penicillins, they usually produce acids and stuff which break these down, and with the help of R group, you might be able to be resistant to it. Now, moving on. So this whole compound here is known as the ß-lactam. This whole thing, so the ß-lactam ring, as well as this here, the whole thing is known as the ß-lactam, and here is the R group attached. So you can see that the now before we get into a bit more about what penicillin is, we need to talk about something also known as peptidoglycan. Peptidoglycan is basically a fancy way of saying cell wall, but we have eukaryotic cells, which are human cells, and prokaryotes, which have bacterial cells. So these ones have a cell wall, whereas these ones have a peptidoglycan, and it's essentially the same thing as a cell wall. This is found in human somatic cells, and this is found in bacterial cells. They have a peptidoglycan instead of having a cell wall. It's essentially same thing as cell wall. Now, peptidoglycan is what we're going to look at because we're going to concentrate on that. It's essentially formed from monomers of sugars called monomers of sugars. So you can see here on the bottom I've written it out. Peptidoglycan is a polymer consisting of peptidoglycan monomers. Each peptidoglycan monomer consists of 2 sugars and a tetrapeptide. Glycosidic bonds are the ones that connect the monomers together, and the peptides interlink it via transpeptidase enzyme. So first, the steps in order to create a peptidoglycan is the monomer synthesis. So what you need is it happens in the cytosol. Then it's polymerization. So this is the addition via glycosidic bonds, and then you have cross-linking, and we'll look into a bit more detail. And this happens in certain layers. So the two monomers that we want to be aware of is N-acetylmuramic acid and N-acetylglucosamine. So we call it NAM or NAM and NAG, as long as you can remember it as that. So the thing that you want to look out for here is that NAG and NAM are formed via the glycosidic bond, which is shown there. Similarly, the key part is this part here, is the pentapeptide. Right, this is the pentapeptide that I was referring to, which I accidentally got wrong by saying tripeptide. Sorry, it's meant to be a pentapeptide, so 5 molecules. And these then go on to, so the NAM joins to the NAG via glycosidic bond, and then so on and so on and so on. And these peptides also build up on the bottom. What happens in phase 3, it's known as cross-linking of these peptides. So this one will cross-link to the adjacent peptide via transpeptidase enzyme. And this will form a peptidoglycan bridge, right? And that's what that bond there between the pentapeptide is what ensures structural integrity and really keeps the peptidoglycan, the cell wall of the prokaryotes or the bacteria, to become very strong. Now, you might have heard from A-level or biology teachers or anyone that antibiotics usually work in a sense that they inhibit this transpeptidation, so the cross-linking between the adjacent peptides, which then mean the structural integrity is not maintained, and this peptidoglycan cell wall becomes less strong, so water can enter and then die due to osmolysis. Yeah, so that's basically what happens. So the two key sugars are NAM, N-acetylmuramic acid, and N-acetylglucosamine. Right, now we have two types of bacteria here, Staphylococcus aureus and E. coli. So gram-negative, this is gram-positive Staphylococcus aureus, and gram-negative. The main difference here between these two is this molecule here, mesodiamelic acid, whereas this has a lysine. Right, that's the only difference. But why this is gram-positive and why this is gram-negative, we'll look in a moment. Now, as I've already mentioned, composed of cross-linked chains of peptidoglycan monomers. So this is the main thing that you want to look at. It's made from an NAM and NAG, which usually has a pentapeptide. This then binds on to another NAM, NAG, so another pentapeptide. So in theory, what you have here, these are the glycosidic bonds, right? So these are the glycosidic bonds, and you form a bond between the peptide. This is the cross-linking, and this is catalyzed by the enzyme transpeptidase. So you can see in here on the left, it's transglycosidase enzymes join these monomers together to form a chain by glycosidic bonds, and transpeptidase enzymes then cross-link. So this one's catalyzed by transpeptidase, so you can tell from the name days, and these ones are, the glycosidic bonds are catalyzed by transglycosidase. I hope you get the idea what I'm trying to say. Okay, so moving on. Now you can see here penicillin blocks transpeptidase enzyme. So penicillin usually acts on these ones, so blocks enzyme. So if it blocks it, then that means you can't form this bond here, so therefore cross-linking cannot take place. So as you can see here, there's the NAM, NAG, so something similar to what I've drawn out here, and there's the bottom part. So there's one transpeptidase here and the other one. So this pentapeptide here cross-links with this one, and this here is the bond, the peptide bond between the two pentapeptides, forming the cross-link. Now, if you have an antibiotic, what it does is it usually stops this bond from forming, this bit here, which means there's no cross-linking between these adjacent layers. So it makes the peptidoglycan cell wall much weaker and more prone to osmolysis. Now, a better diagrammatic view of this would be penicillins block transpeptidase enzyme and prevent formation of peptide crosslinks. So you can see the bond is formed between lysine and alanine here, right? So NXR glucosamine, NAM, pentaglycine bridge. So this bond here is also known as, so you can call it a peptide bond, or to be more specific, it's called a pentaglycine bridge, and it's catalyzed by a transpeptidase enzyme. DLA, the alanine eliminated during transpeptidation. So one of these, number 3, will be eliminated. So this one always gets removed once transpeptidation, so once this bond is formed. But for now, all you need to know is that there's this bond between the pentapeptides, which forms a cross-linking. Now, penicillins are beta-lactam stability. So there are, as you know before, this ring here is a beta-lactam. This is the thiosylidine ring, but we're more interested in this. Certain bacteria have developed resistance to antibiotics in a sense that they create beta-lactamase, which is an enzyme created by them, which break down this beta-lactam ring. If it breaks down this beta-lactam ring, it basically means the penicillin becomes ineffective, really. So examples of naturally occurring penicillins include benzyl penicillin, procaine, which is a solid version of it, phenoxymethyl penicillin. Benzyl penicillin is usually naturally occurring, but the problem is it's broken down by beta-lactamases. It has very poor absorption due to it can be easily broken down by acid or by the absorbed food. So the bioavailability is really low. So it's in fact given by an injection rather than given orally. Procaine is a solid version and is usually known to be effective for about 24 hours post-administration. So it's usually given as a treatment post-surgery or prophylaxis, basically. So a post-antibiotic effect once post-operation or something to prevent further encounters of any bacteria. Phenoxymethyl penicillin, similar spectrum to PCG but less potent, acid stable, can give orally but absorption is variable. Now, beta-lactam, broad spectrum beta-lactam resistant penicillins include, so if I was to get rid of this, so if we make a note of the different types of penicillin we have as we go along, much easier. So we have natural beta lactamase resistant. And we have something called broad spectrum. Now, natural occurring was the previous slide. So benzyl penicillin, procaine. So just give procaine, benzyl penicillin. The beta lactamase resistant ones include flucloxacillin, which is the most important one, or given by doctors. Flucloxacillin. You also have ficillin, methicillin, which is acid liable, which means it can be broken down by the acid in the gut. So it's usually given by an injection by beta-lactam resistant. So I'll put that in red. So you got flucloxacillin, cloxacillin, nafcillin, now the main one is methicillin. It's much more stronger, and but because it's acid liable, it's usually given via injection, so intravenously. Now the Cmax of all of these, it's about 1 hour, and the half-life is about 30 to 1 hour, 30 minutes to about 1 hour. Now if we carry on going forward, so that's beta-lactam resistance. Now you have broad spectrum, is something that we might have taken when we fell ill or something. So we have ampicillin, or the most common one is what is given or prescribed is amoxicillin. And then we have extended spectrums of penicillins. So usually include carbonicillin, ticricillin, and the reserved ones are like very potent against more potent versus gram negative than gram positive. So these ones target especially gram negative ones. So the two most familiar ones should be ampicillin and amoxicillin. Now, taking these drugs, doesn't always mean it's a beneficial effect. Well, it has a potential to clear the bacterial infection, but it also has some sort of nasty side effects like skin rashes, anaphylaxis. It can cause certain allergic reactions. So here you see loss of skin pigmentation. Now, usually when you end up going to a doctor or a healthcare professional and then they turn around to you and say you have some sort of bacterial infection, right, they first treat you upon your basis of your symptoms and stuff like that. So they want to identify if it's a viral, if it's a fungal, or if it's a bacterial infection. So usually what they do is they're trained enough to identify between those three, so they'll differentiate between, so what type is it? So it can be fungal, bacterial, or it can be viral. So once they come to a decision of which one it is, so from the symptoms and different types, then you have different types of bacteria, different types of fungi, and different types of viral. So So beta-lactams are effective against certain types of bacteria like Streptococcus. There is Staphylococci. There is pneumonia, Neisseria, and various. There's loads of classes of different bacteria and different viral and fungal infections. So an example here is beta-lactams usually target the cell wall peptidoglycan, and these are beta-lactams are beta-lactamase resistant, so they're resistant to the degradation from the beta-lactamase produced by bacteria. So bacteria has no resistance against these, well, at least for now. Polymyxins usually go against the outer membrane. Then you have something, so these ones are also antibiotics, so they are also different types of antibiotics. So but beta-lactams are probably the most commonly used ones. Polymyxin is not that commonly used, but cell membrane drugs, the antibiotics that target the cell membrane, are isoniazid. This is prescribed as a first-line treatment for tuberculosis. Sulfonamides used in the cytoplasm. Fluoroquinolones, like antimalarials, like what they do is they inhibit chromosome and DNA synthesis as well as protein transcription. So therefore these stop the bacteria from dividing further. Now tetracyclines and macrolides, they affect the ribosome synthesis. So fairly from here you can see that antibiotics can be classified into various different families. So if I was to draw that out, so you can see that you have beta-lactams, you got polymyxin, you got isoniazide, you got fluoroquinolones, got sulfonamides, tetracyclines, and macrolides. Right, so these are different ones. So from here you can tell that beta-lactams target the cell wall, so the peptidoglycan. Polymyxins, outer membrane. Isoniazid, the cell membrane, so the inner membrane, so this bit here. Fluoroquinolones target DNA and DNA synthesis. Sulfonamides target the cytoplasm, and tetracyclines target our ribosome synthesis. So ribosomes are involved in making proteins. They're also involved in transcription. So if indirectly affects transcription as well. Now, going forward, protein synthesis is really important, and if you're not aware of what protein synthesis is and the whole procedure behind it, you might want to check out a video on YouTube. And there's a link provided in the lecture slide. So the main thing is you need to be aware is in protein synthesis we have different phases, so different stages. So first what happens is you've got your DNA, it's split in, so it's a semi-conservative model. it's the hydrogen bonds between the nucleic acids is broken first, which then one of them acts as a template strand, which forms the RNA. The RNA is then acts as a template strand to which ribosomes bind, in which tRNA with anticodon sequence binds to the complementary codon on the RNA. This is then replicated and formed into DNA and then spliced, so it removes introns, and then leaves behind exons. So these exons can then be shuffled up to form various different variations and so on. So the main two ribosomes that we're interested in are 30S and 50S, and you should always know that mRNA is translated from the 5' end to the 3'. So this priming is related to the sugar over DNA backbone, which I'll go through in various different lectures on DNA synthesis and protein synthesis. So the key molecule here that you want to be aware of is something called N-formylmethionine, so FMET. This usually initiates the process of tRNA binding into the A site, P site, and then the next tRNA coming along and binding onto the A site, and then subsequently the first one moves into the E site, which is the exit zone. Go into a bit more detail as we go through the slides. So at each cycle, there's 3 sites present on the ribosomes. So A is usually empty. P contains the peptidyl tRNA, so it contains the amino acid and the anticodon. The E is the exit site. So if you go over to this website here, and you don't really understand what protein synthesis is, you can have a quick look at the video, because this video is not based on that, but I'll still touch upon it. So initially FMET controls the process, then elongation takes place, and then termination and disassembly. Now mRNAs, ribosome and tRNAs, the 3 compounds that you need. Right, so initiation is usually blocked by these type of antibiotics, aminoglycosides and tetracyclines. Right, you also know chloramphenicol block transpeptidation, and macrolides are involved in blocking translocation. So transpeptidation is basically forming the peptide bond between the amino acids, whereas translocation it stops the assembly of the so it prevents the tRNA from entering the E site, which is the exit site, so therefore it can't be removed. So aminoglycosides are another example of antibiotics. So a key example of this is streptomycin. It was the first aminoglycoside characterized, and what these guys do is they cause misreading, and they also interrupt and prevent initiation from taking place. So if you go back to the slide, you can see aminoglycosides are involved in blocking initiation of protein synthesis, as well as tetracyclines and chloramphenicol prevents transpeptidation from taking place. So an example of aminoglycoside is streptomycin. Gentamicin is also an example. These are given parenterally, so they're given via injection. They're broad spectrum. They're bactericidal, so this means bactericidal basically means that once they have an effect, they basically kill off the bacteria, so it stops it from further replicating, and then it causes death of the bacteria too. Tetracyclines, as you already saw, also affects the initiation. So in here it inhibits the amino acyl tRNA, so binding to the A site on the ribosome. One way to identify if patients taking tetracyclines is usually you can see it is deposited in the bones/teeth, so you get this bluish greenish color underneath your teeth, and the absorption is reduced by milk. So patients usually prescribed tetracyclines are told to avoid consuming milk. Chloramphenicol it inhibits transpeptidation. So this is the process by which. So initially, what happens is your first tRNA. So here you have this is the 50S, this is the 30S. Here is the mRNA. On top of the mRNA what is contained is known as codons. So every three nucleotide makes up one codon, and these tRNA contains anticodons which match to them. The only difference is that mRNA has a uracil, and so does a tRNA. Now, once these bind, so they come with a amino acid joined to them. Now, the next one comes into the A site. Right. So from the A site, what happens is a peptidyl transferase causes a peptide bond between these two amino acids, and then this one is then the bottom part is then eliminated into the E site, and then it's disassembled. The A then further moves on to the P site, and then to the E, and then assembled like that. What happens to the top bit is the amino acid keeps on racking up. So chloramphenicol prevents transpeptidation, so it prevents the binding or the bond formed between the amino acids. Side effects of chloramphenicol, it's a bacteriostatic drug, so in a sense that it doesn't kill off the drug, all it does is prevents replication of the drug. It's potentially seriously toxic. In newborn babies, we have a syndrome called grey baby syndrome. It can cause idiosyncratic reactions, so like skin abscess, skin burn, and various different types of unwanted side effects. Macrolides, on the other hand, inhibit translocation. So erythromycin, clarithromycin, azithromycin, these all bind to the 50S subunit and inhibit translocation. So it basically blocks the exit tunnel, so the E site. Now, so if you want to quickly go back, so initially we have something like this. So we have the beta-lactams, then we have tetracyclines and macrolides, chloramphenicol, aminoglycosides. Examples: streptomycin, gentamicin, tetracycline, chloramphenicol. Now, so for example, what happens is they collect a data or a biopsy from your infected tissue, and then they send it off to the lab. The lab technician then undergoes various different processes by which he does various different tests by which he identifies what type of bacteria is present in the patient. So once he's identified it, for example, if it's Haemophilus influenza or Helicobacter pylori, which is involved in causing ulcers and stuff, what is administered is an antibiotic called clarithromycin, which is a macrolide. So it prevents. What it does, it prevents the translocation, and it can either be bactericidal or bacteriostatic. So bactericidal means it can either kill it off or at least stop it replicating. So it really depends. So some of these drugs can act on the same species of bacteria as well. So, but it's mainly identified via which species is present. So therefore, you can give that type of drug which is most effective in suppressing that certain species that's causing the harm. Linezolid, so oxalisinodone is a new class of antibiotic that's recently been in the media and been developed. So linezolid is first to be introduced, and it's meant to be a variety of gram-positive bacteria. So mainly against MRSA. So MRSA stands for methicillin-resistant Streptococcus aureus. So if you remember back, methicillin is a type of not broad spectrum, ß-lactam drug, which means this MRSA, so the Streptococcus aureus here, is resistant to methicillin. So even when it's given by IV injection, it adds some sort of resistance to this compound. So that's why those bacteria are called methicillin resistant aureus. And the drug that they've recently invented called linezolid is meant to have cure against MRSA. Now, resistance, you might be thinking, where do these, it's always all over the media, where does resistance come from? How do these bacteria develop resistance? They don't have a brain like humans, then how is it possible? Well, resistance cannot be, there's different types of resistance. So you have inherent resistance, acquired is then split into vertical and evolution. So the main one is inheritance. So bacterium has an outer membrane that establishes a permeability barrier against antibiotics. So this has always been there. So it's genetically inherited and resistant. So in gram-negative bacteria, they have an outer layer formed from polysaccharides and polypeptide. The acquired resistance is something that's acquired, so it's gained once they're in an environment. So this is driven by a process known as mutation and selection. So there could be a mutation in a gene, which means it's now resistant. The protein that is produced is different, which means that it's more resistant to that particular drug. This can then be passed on vertically, so to a member of bacterial population, or it can be passed on exchange of genes via horizontal evolution. So this is via a process called conjugation, in which a tube forms between two bacteria and they send off the circulate DNA, so plasmid, and then that goes into the other corresponding recipient cell, and then they take on board the gene for resistance. So vertical evolution is simply being replicating and dividing and passed straight on. Horizontal is when you have two cells like this, and they form a tube. So this is a conjugating tube or a tunnel. This is your circular DNA, which contains resistant to tetracycline and various different types of. This is your areas within your gene which contains genes. So once it becomes here, these also gain it. So now these become resistant to. These ones were the initial parent, or the initial, you could say initial, and this is the recipient. So this is known as horizontal. So it's going across the x-axis, so going across. Now vertical is simply the opposite. So it's the cell with a circular plasmid. Now that then goes on to divide the different cells. which also similarly has the resistance, which is indicated here by the red bit. Okay, so this is vertical, this is horizontal, and this is vertical. Now, let's move on. So it can be inherited resistance, so such as gram-negative bacteria, which means they've always will have it, or it can be acquired once they're present in an environment via mutation and selection. Now, gene transfer. So bacteria exchanges in genes in nature by three processes: conjugation, transduction, and transformation. Conjugation is what's shown here. So you see, involves cell-to-cell contact as the DNA crosses sex pilots from a donor to a recipient. So here's the resistant recipient, and here's a donor. Now, and here's the conjugating tube. Transduction is when a virus transfers the gene between a bacteria. So a viral cell, so let's see, viral cell gives it off to a bacterial cell. In transformation, DNA is acquired directly from the environment, having been released from another cell. So transformation is when you release it to the environment and then these cells pick it up, whereas transduction is a viral cell comes along and passes it on, and conjugation is when two bacteria interact. So conjugation, a simple way to remember is conjugation is conjugating, so it's coming together. So two bacteria. Transduction is passing on, so it's giving it off from somewhere to another area, so transducing. So in physics you will learn transduction and transducing agents. So it's changing positions from one to a complete new environment. So it's going from a viral environment to a bacterial environment. And transformation is when they're directly released into the environment and it's picked up by subsequent recipients. Here's a photo, so you can look at that. Now, there's biochemical mechanisms involved with resistance. So, for example, chlamydia, it's a sexually inherited disease, sexually passed on disease. And what you have is there's an absence of the target, so there's no peptidoglycan. So if there's no peptidoglycan, where do these ß-lactam antibiotics act? Because they act on peptidoglycans, right? So clearly it makes that whole class of antibiotic completely ineffective. Now, target inaccessible is things like with gram-negative bacteria usually has a very thick outer membrane, which means that drugs cannot easily enter it. Inactivation is another process. So by this it means is that the beta-lactamase's enzyme breaks down the beta-lactam ring, which makes the antibiotic inactive. So it makes the beta-lactam inactive, aminoglycosides, chloramphenicol inactive. Modification of the drug sensitive site. So there's modification of the drug where the drug binds. And loss of permeability or increased efflux protein. So if you have a very increased efflux protein, it's very hard for the drug to get into the cell because these efflux protein would simply take the drug back out. So 1, you have target absence. 2, you have inactivation. 3 is target inaccessibility, so outer membrane. Modification of the drug's binding site, so it's not complementary anymore, so therefore the drug won't bind. Loss of permeability or increased efflux. So this is basically a table which shows all the different examples. So you can see chloramphenicol. You know that chloramphenicol inhibit transpeptidation, reduced uptake into cell. So the process is it has very high efflux proteins, therefore they decrease the uptake of these antibiotic tetracyclines. They are involved in blocking initiation. They are active effluxed. SS-lactams, erythromycin, denomycin, eliminates reduce. So if you want to have a read across those. Antibiotics may be enzymatically degraded into an inactive form. The ß-lactam ring is, so we know this. So Staphylococcus have routinely been associated with production of beta-lactamases. So what you would administer these ones, so Staphylococcus would be beta-lactam. So beta-lactamase resistant drugs, antibiotics. Drugs were made to resist the activity of staphylococcal penicillinase. They had side chains that prevented the staphylococcal beta-lactamase from binding to the antibiotic and hydrolyzing it. Methicillin is a staphylococcal beta-lactamase stable penicillin, was introduced in clinical practice in the 1960s. So resistance MRSA is a really touched upon topic in the modern society. And what MRSA stands for is methicillin resistant Staphylococcus aureus. So the bacteria that's present in these infection is Staphylococcus aureus, and they're methicillin resistant. So resistance is due to the temperature sensitive production of an extra penicillin binding protein, so PBP, penicillin binding protein. This is insusceptible to the inhibition of methicillin. Now, methicillin also produces beta-lactamase, but beta-lactamase is ineffective against methicillin. So what they're saying is the key thing is this penicillin binding protein that is produced by the Staphylococcus aureus, which is preventing the methicillin from acting on these bacteria. And now the only drug that they've recently discovered in order to get rid of or terminate MRSA is vancomycin. So vancomycin is usually administered to people who is thought to have been affected by MRSA. Now, the difference is normal penicillins, right? They block the pentaglycine bridge. So this is the pentaglycine bridge. So they prevent transpeptidation from occurring. So therefore, there's no cross-linking. Right? Vancomycin binds with the substrate, not the enzyme. So here. So most penicillins usually block the transpeptidase enzyme, which catalyzes this formation of here. So they don't directly go and affect this, they affect the enzyme which forms these bonds. Now the difference is vancomycin binds with the substrate, not the enzyme, in contrast, which penicillin inhibits peptidoglycan synthesis. It binds to the alanine and then prevents transglycosylation, steps in, so it prevents glycosidic bonds from being formed in cell wall synthesis. This prevents peptide bonds, but vancomycin prevents glycosidic bonds. So that's the key difference. If you go back, resistance fighting back. Beta-lactamase inhibitors are proteins designed to inhibit. These inhibitors, carbonic acid, ß-lactam, tosbactam, functions by binding to the beta-lactamase enzyme. So it says here that how the modern medicine has been evolved is by a process, and they've tried to fight back resistance. So beta-lactamase inhibitors are proteins designed to inhibit and destroy the effectiveness of beta-lactamase enzymes. Inhibitors generally have a little antimicrobial properties themselves, and so are combined with the beta-lactam antibiotic. So these type of acids, and these are added on to the as a side group. So if you remember before, I mentioned that the R group is really important, right? And what happens is acids like calvinic acid, ß-lactam, tazobactam, these are all added onto the R group, and this R group then determines what class it is. So if things like these are added, then they become a beta-lactam resistant drug, lactamase resistant drug, so beta-lactams, and they then help in the breakdown of beta-lactamase enzymes, which is produced from the bacteria itself. Hope that makes sense. Now, antibacterial combination of amoxicillin, so Augmentin is this molecule. So you have your beta-lactam ring here, this NH, and all of this here, it's the side group. So up to here is the amoxicillin. So amoxicillin is shown here. So antibacterial combination of amoxicillin and the beta-lactamase inhibitor potassium carbonate. So here. So amoxicillin is a broad spectrum, if you remember, but that means it doesn't contain the enzyme, or it doesn't contain the side group, which stops the beta-lactamase from breaking the amoxicillin down. Now, antibacterial combinations, by that sense, you can add amoxicillin and potassium carbonate, which is a beta-lactamase inhibitor. Carbonate acid is produced from fermentation of Streptomyces. So this acid is produced from another bacteria, Streptomyces clavularis. From here, this is where it gets the name covalent. Beta-lactam structure related to the penicillin inactivates. So yeah, so that's the end of that lecture. Hope you found it useful, and see you in the next lecture. Please make sure to leave a comment, like, and subscribe to the channel.